Every second of your life, billions of tiny power stations inside your body are working non-stop to keep you alive. When you blink, walk, think, or even breathe, energy is being released at the cellular level. But have you ever stopped to wonder where this energy actually comes from? Food gives us energy, yes, but food itself must be broken down and converted into a usable form inside our cells. This is where one of the most powerful and fascinating processes in biology takes place: the electron transport chain, hidden deep inside the mitochondria. This microscopic system uses moving electrons to generate most of the energy your body needs. It may sound complex, but at its core, the electron transport chain is like a biological power plant, carefully designed to extract energy efficiently. In this video, we are going to break down the electron transport chain in a simple and clear way, connecting it to cellular respiration and explaining why it is the final and most important stage of energy production. We will explore where the electron transport chain occurs, how electrons from NADH and FADH travel through a series of carriers, how oxygen plays a critical role, and how this movement of electrons ultimately leads to the formation of ATP, the energy currency of the cell. By the end of this video, you will understand how your cells turn chemical energy into usable power, and why the electron transport chain is essential for survival in all aerobic organisms. What is the electron transport chain? The main role of the electron transport chain is to transfer high-energy electrons from molecules such as NADH and FADH to oxygen. These molecules are produced during glycolysis and the Krebs cycle and act as electron carriers. They donate their electrons to the electron transport chain, starting a flow of electrons through different complexes. As electrons move along the chain, they lose energy gradually. This controlled release of energy is extremely important because it prevents energy from being wasted or released all at once. Instead of being used directly, the energy released from electrons is utilized to pump protons, or hydrogen ions, across a membrane. This creates a difference in proton concentration, which later drives ATP synthesis. Oxygen plays a critical role in this process by acting as the final electron acceptor. Without oxygen, electrons would back up in the chain and ATP production would stop completely. The electron transport chain is considered highly efficient because it produces the largest amount of ATP compared to other stages of respiration. It is also a key reason why aerobic respiration is far more effective than anaerobic processes. In simple terms, the electron transport chain is the energy harvesting system of the cell, converting the energy stored in food into a form that cells can immediately use to perform life-sustaining activities. Location of the electron transport chain. The electron transport chain takes place inside the mitochondria, which are often called the powerhouse of the cell. Mitochondria are double membrane structures, meaning they have an outer membrane and an inner membrane. The electron transport chain is specifically located on the inner mitochondrial membrane, making this location essential for efficient energy production. The inner membrane is folded into structures known as crista, which greatly increase its surface area. This increased surface area allows more electron transport chains and ATP-producing enzymes to be present at the same time, boosting the cell's ability to generate energy. Embedded within the inner mitochondrial membrane are a series of protein complexes and mobile carriers that make up the electron transport chain. These complexes are arranged in a precise order to ensure the smooth transfer of electrons from one carrier to the next. Between the inner and outer mitochondrial membranes lies a narrow space called the intermembrane space. This space plays a crucial role in ATP production because it is where protons accumulate during the electron transport process. The matrix, which is the innermost part of the mitochondrion, contains enzymes for the Krebs cycle and also serves as the source of protons used in the chain. The location of the electron transport chain is not random. The inner mitochondrial membrane is selectively permeable, meaning it tightly controls the movement of ions, especially protons. This control allows the cell to create and maintain a proton gradient, which is essential for ATP synthesis. If the electron transport chain were located elsewhere in the cell, this gradient could not be maintained and ATP production would be inefficient. In organisms that lack mitochondria, such as prokaryotes, the electron transport chain is located in the plasma membrane. This shows that, while the structure may vary, the principle remains the same. The strategic placement of the electron transport chain ensures maximum energy production and makes it one of the most important cellular systems for life. Step-by-step working of the electron transport chain. The working of the electron transport chain begins when NADH and FADH donate their high-energy electrons to the chain. NADH releases its electrons to the first protein complex, known as complex I, while FAD donates electrons to complex II. From there, electrons move through a series of carriers, including ubiquinone and SUT, passing through complex III and finally reaching complex IV. As electrons move through these complexes, energy is released in small, controlled steps. This energy is not used to make ATP directly. Instead, it is used to pump protons from the mitochondrial matrix into the intermembrane space. This proton pumping occurs mainly at complexes I, III, and IV. As a result, a high concentration of protons builds up in the intermembrane space, creating what is known as a proton gradient. This gradient represents stored potential energy. Similar to water held behind a dam, the difference in proton concentration and electrical charge across the inner membrane creates a strong force that drives protons back into the matrix. However, protons cannot pass freely through the membrane and must move through a special enzyme called ATP. At the end of the electron transport chain, oxygen acts as the final electron acceptor. Oxygen combines with electrons and protons to form water. This step is extremely important because without oxygen, the electron transport chain would stop functioning and ATP production would cease. This is why oxygen is essential for aerobic respiration and survival. The step-by-step movement of electrons ensures that energy is released gradually and efficiently, preventing damage to the cell. This precise and organized process allows cells to generate large amounts of energy safely and continuously. ATP synthesis and importance of EXA. ATP synthesis occurs through a process called chemiosmosis, which is directly linked to the electron transport chain. The proton gradient created by electron movement stores energy that is later used to produce ATP. Protons flow back into the mitochondrial matrix through ATP synthase, a large enzyme embedded in the inner mitochondrial membrane. As protons pass through ATP synthase, the enzyme rotates and uses this energy to convert ADP and inorganic phosphate into ATP. ATP synthase works like a molecular turbine, powered by the flow of protons. This mechanism makes ATP production extremely efficient and allows cells to produce a large amount of energy from a single glucose molecule. Most of the ATP generated during cellular respiration is produced at this stage, highlighting the importance of the electron transport chain. The electron transport chain is essential not only for ATP production, but also for maintaining metabolic balance in the cell. It regenerates NAD and FAD, which are required for glycolysis and the Krebs cycle to continue. Without the etc., these earlier stages would stop, and energy production would collapse. The importance of the electron transport chain becomes especially clear in oxygen deficiency. When oxygen is unavailable, electrons cannot be transferred, and ATP production drops drastically. This is why cells shift to less efficient anaerobic pathways under oxygen conditions. In summary, the electron transport chain is the backbone of aerobic respiration. It ensures efficient energy conversion, supports continuous cellular activity, and makes complex life possible. Without it, organisms would not have the energy needed to survive, grow, and function. As we reach the end of this video, it becomes clear why the electron transport chain is considered the heart of cellular energy production. This remarkable process, taking place inside the mitochondria, shows how living cells convert the energy stored in food into ATP, the fuel that powers every activity of life. From the movement of electrons through a series of carriers, to the creation of a proton gradient, and the final synthesis of ATP. Each step of the electron transport chain is carefully coordinated and highly efficient. It also highlights the vital role of oxygen, without which this entire system would come to a halt. Understanding the electron transport chain helps us see that energy is not created suddenly, but generated through precise molecular events happening every moment inside our cells. Whether it is muscle contraction, nerve signaling, growth, or repair, all depend on the ATP produced through this process. By learning how the electron transport chain works, we gain a deeper appreciation of how life sustains itself at the microscopic level. If this explanation helped you understand cellular respiration more clearly, don't forget to like the video, share it with others who are studying biology, and subscribe to the channel for more easy-to-understand science content. Remember, when you understand energy at the cellular level, you begin to understand life itself.